learned a lot about influenza in pigs since the 2009 H1N1 pandemic, particularly in Mexico. Many of you are probably familiar with this idea that pigs are mixing vessels for pandemic influenza viruses, that they can be infected from avian viruses and human viruses, that influenza viruses have these segmented genomes, and they can swap segments through reassortment and create novel viruses in pigs that can then transmit to humans and cause pandemics. This was evident during the 2009 H1N1 pandemic, where pigs served as mixing vessels. Basically, in this case, you had two very divergent swine lineages. Here on the left, this is the Eurasian swine lineage that's found in Europe and parts of Asia. And then you have a very different North American swine lineage that's in North America and also in parts of Asia. And you had a reassortment event between these two divergent lineages within a pig that then transmitted to humans and caused the first outbreak in Mexico. And at the time, we were able to do a lot of sequencing and very much sort of characterize where this virus was, where all the different segments came from, what lineages these were. There's a very sort of deep evolutionary history of how they came from different birds and reassortment events, but we had no idea where this particular reassortment event actually occurred, which country. And it was sort of a mystery at the time because you had the first outbreak in humans in Mexico. That was quite clear from the data that we got from humans, that at the time Asia was the only country, or the only region in the world where we knew that both North American and Eurasian swine lineages co-circulated. And there was some early work right at the onset of the pandemic from Gavin Smith's group showing that there were viruses collected from Hong Kong that were the most phylogenetically related to the human pandemic viruses, which are here in the top. And this was sort of, supporting maybe that the virus came from pigs in Asia and somehow started the first outbreak in Mexico. But it was noteworthy that at the time of the onset of the pandemic, there were 0 sequences from pigs in Mexico available on GenBank. There were major gaps in sequencing globally from pigs, and it was really it highlighted how little we knew about the global evolution and ecology of influenza in pigs at the time. So since 2009, we've really ramped up surveillance in pigs globally for influenza viruses. And one of the things that's become apparent, and I'm not going to talk about it that much, but we recently sort of did a review highlighting it, is how much humans are transmitting influenza viruses to pigs, and this idea of sort of reverse zoonosis. And we were able to characterize, so after the pandemic emerged in humans, we were able to identify humans transmitting the pandemic virus back to pigs on 6 different continents, hundreds of times in the United States, just a really fluid interface from humans back into pigs. And these viruses are continuing to circulate in pigs and reassort with others and have been really sort of creating more genetic diversity in pigs. We also were able to go back in time, all the way back to the 60s and 70s, and see that this has been going on for decades, human seasonal viruses jumping into pigs, becoming established, and this is really a major source of genetic diversity for flu in pigs. Another thing we've been looking at as we characterize viruses all around the world is I'm going to blow your mind here, the pigs fly. They go from North America to Asia, Europe to Asia, long distances, Canada to Russia. That's what this little box is here. That's going on about 1,200 pigs going on a Boeing from Canada to Russia. And we can sort of trace how these pig movements that are very long distance are actually spreading the virus all over the world and are really important in understanding sort of how populations are mixing and sort of spreading lineages all over the world. So these are Google Earth visualizations inferred directly from genetic sequence data. These are the Eurasian lineage spreading first into Thailand and then in the early 2000s into China. So multiple introductions. So even though you have the same virus in Thailand and China, they actually represent independent introductions from Europe. North America is also a major source of viruses around the world. Vietnam, Japan, Korea, also China, and as we'll see later, also Mexico. So we can do sort of a generalized linear model to test specific predictors of what's driving the spatial spread of influenza around the world, and we looked at all different kinds of things like geographical distance, swine population sizes, and and also the volume of live swine trade between countries. And you probably can't read these, but this is the volume of live swine trade pairwise between different countries, and that was consistently the strongest predictor of how the flu is spreading. So it's this trade of live pigs. Since we don't have sequence data from all countries, but we do have a lot of trade data, we then used an SIR model to predict from a given country here in black where its influenza viruses would spread globally based on our knowledge of what its trade partners are. So you can look at the United States, for example, see the viruses would spread long distances to China and Russia and many parts of the world in Latin America, similar with Canada below. European viruses will also spread eastward, but then you can identify these, what we call sink regions, like China and Mexico, countries that actually don't export pigs to other countries. For China, it's very strategic to have a stockpile of pigs, and so it's not, even though 500 million pigs are in China, it's not a source of viruses in neighboring countries like Vietnam and Thailand. They're actually, many of the viruses there were independently imported from Europe and North America. So there's this very sort of rich, complex ecology based on the live swine trade that we can begin to tease apart. So then in a collaboration with Mount Sinai Group, with Adolfo Garcia-Sashvin and his postdoc Nacho Mena, we looked at influenza viruses in Mexican swine, and we finally resolved sort of the origins of this 2009 H1N1 pandemic. So Eurasian swine viruses had never previously been identified in the Americas. That's why we thought maybe the pandemic virus came from Asia. But here we actually, with new surveillance, particularly from central Mexico, we found that there were Eurasian viruses circulating in Mexican pigs. Not only that, they were reassorting with North American lineages and creating the exact same genotype that caused the 2009 pandemic. And lastly, they were also very closely related to the human pandemic viruses that we isolated in 2009 for at least 7 of the segments. So this sort of, we eventually said this is solid evidence that this virus did emerge from pigs in Mexico, spread from pigs to humans in Mexico, and better understand the origins of the 2009 pandemic. One thing that, in addition to Mexico having a lot of genetic diversity, so it's importing viruses from North America, from Europe, and they're all reassorting. It's also, we've been sort of, through another collaboration with people in the University of Minnesota, we've been looking at sort of the spatial heterogeneity of swine production within Mexico. And unlike the U.S., where you have a lot of movements and a lot of mixing, in Mexico you have actually very different dynamics and different viral populations in different parts of the country. So if you look more closely within Mexico, there's the Sonora region, and they primarily import viruses across the border from Texas and Oklahoma and the major swine population there. If you look at the central region, where we found the sort of pandemic-like viruses, they get viruses from North America and also from Europe, and that's what allowed those lineages to mix and reassort. And then we also did a more intensive study of the southeastern region in the Yucatan Peninsula, and looked at sort of, and found that there's actually extremely high genetic diversity in this region as well. And that's because there's two things going on. There's viruses that were in the central region that we identified in the previous study have spread into the Yucatan through pig movements. And also there's been lots of human to swine introductions in Yucatan, including a seasonal human virus from the mid-1990s that's been circulating for decades there, as well as multiple introductions of human 2009 H1N1 pandemic viruses. So you can look at another region of Mexico has become sort of a hotbed for genetic diversity for influenza. So just in conclusion, we identified exceptional genetic diversity in pigs in Southeast Mexico. We can interpret how this got there by studying pig movements within the country, by understanding the global live swine trade and how viruses are being spread intercontinentally between Europe to Mexico, and also understanding all the millions of pigs are being moved from North America to Mexico and spreading viruses multiple times with them. This idea of reverse zoonosis is really important, and humans are a major source of influenza in pigs globally, and including in Southeast Mexico. and genomic reassortment. It happens very easily in pigs. That's why they're considered this mixing vessel. They can harbor lots of different lineages, unlike humans, and allow different lineages to reassort and create new combinations, some of which have pandemic potential and which sort of merit further analysis experimentally. So we thought sort of the implications of this. So things like quarantine and influenza isn't regularly tested when pigs are shipped internationally. Also thinking more about vaccination of swine workers and looking at this human-animal interface from the opposite direction and recognizing that we also have to pay attention to humans giving viruses to animals. We've also seen this not just in pigs, but in lots of other species. And also in sort of understanding surveillance, the more we can sort of understand this global ecology of influenza in pigs, we can better target where we need to conduct surveillance. For example, China is not necessarily a good sentinel for the region of Asia because it's even though it hosts this enormous pig population, its pigs aren't really mixing with pigs in other countries. And same thing for the U.S. At the time, we might have thought that we have very intensive surveillance in the United States, so maybe we know what's in Mexico because they're geographically close, but actually, there can be very different viruses because Mexico doesn't export pigs to the United States, so virus isn't going in the opposite direction. And also in the case of Mexico, it also recognized that there can be a lot of spatial heterogeneity within a country. So even if we've sampled from one geographical region, places where swine production is kind of segment compartmentalized, you actually need to sort of look at many different parts of the country sometimes to get the whole picture.